All right, welcome to this presentation on autosomal DNA. My name is Adam Nisbet, and I'm doing this presentation for the Nesbitt-Nisbet Society of North America. The outline that I have here, we're going to first just do a quick overview of the different types of DNA, in case you're not familiar with different types of DNA. We'll discuss how each of those is inherited, some of the differences between autosomal DNA, which is my specialty and what this talk is about, versus Y DNA, which is kind of the central aspect of the Nesbitt DNA project. Then I'll go into how we can actually use the autosomal DNA for genealogy, who should test autosomal DNA, where to test, and then a few steps that you can take to make your test most useful to our project. So the different types of DNA that we have, I've got a little graphic here that kind of shows the cell, and within that cell you have a nucleus that's got the central portion of the cell, and then in the outer portions of the cell you've got a lot of mitochondria. Inside the nucleus you have 23 different pairs of chromosomes. 22 of them are just numbered 1 to 22. The last one are the chromosomes that determine sex, and so those are the X and Y. Females have 2 X's, males have 1 X and 1 Y. So the surname projects usually use the Y DNA. That's passed father to son, and so it works really well since it usually follows surnames pretty closely. The mitochondrial DNA, which is in the outer portion of the cell, is passed mother to child. And so it traces the maternal line, but because surnames change every generation on that, it's a lot harder to use. So it's usually only used for very special cases where you're wanting to test a theory of someone being connected along a direct maternal line. Autosomal DNA is basically all of those other numbered chromosomes. So you've got a lot of those chromosomes. Those are inherited from all portions of your family, and those you end up being best for doing genetic genealogy of relatively recent generations. So within 5 to 10 generations, if you get beyond around 10 generations, it's hard to use it much. And then finally, there's sometimes what's called xDNA. It's the other chromosome that's part of that 23rd pair. And it's usually tested alongside autosomal DNA, and it's inherited somewhat similarly to autosomal DNA, so it's oftentimes kind of lumped in with autosomal DNA, but it does have a few unique characteristics that are slightly different. So as kind of an overview of where these come from, if we look at a family tree where you've got your paternal side, your maternal side, and all the branches of that, Y DNA only traces the direct paternal line, so only your father's 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 father, and so on. So it's one single line out of that whole tree, and since that's usually the surname, that's why it works so well for surname projects. And it generally gets passed on unchanged, so you can detect matching ancestors many centuries ago. So it's good for tracing deep ancestry along a direct paternal line. Mitochondrial DNA, which is also abbreviated mtDNA, it does the same thing for the maternal line. So it can detect matching ancestors over 500 years ago. But like I said, it changes surnames every generation in most traditional surname inheritance. And so it's a lot harder to make use of for genealogy. Autosomal DNA is inherited from all branches of your family, but it's not inherited equally, and it's only guaranteed to you're only guaranteed to inherit autosomal DNA from every ancestor back around 3 or 4 generations. Once you get beyond that, there's some of your ancestors that you won't have any DNA from, and others you may have 8 to 10 generations or maybe even more, but it starts to fade out, and you never know which branches you're going to have the autosomal DNA from beyond around 3 or 4 generations. So you get differing amounts from each ancestor, and it's distributed all throughout your whole tree. So the autosomal DNA, you get 1 full copy of each chromosome from each parent. So we had the 22 numbered chromosomes. there were 2 versions of chromosome 1, there were 2 versions of chromosome 2, and each parent passes on a semi-random patchwork of their 2 copies. So they had 2 copies, they create a mix of those 2 that's, you know, a chunk of one of them, a chunk of the other one, back and forth, and they do kind of a mix and match reassembly. And because it's semi-random, it's not equally inherited from all your ancestors beyond your parents. So you do get 1 full copy from each parent, but beyond that, you don't get exactly 25% from each grandparent. You may have, you know, 30% from one grandparent and 20% from another. So it's kind of just luck of the draw who you get the different portions of your autosomal DNA from. So there's, because of that, a few key differences in inheritance between Y-DNA and autosomal DNA. So I've kind of highlighted some of these in a side-by-side comparison. So Y-DNA generally passes on unchanged. So since there's only 1 copy per person, that entire copy gets passed on to each son, and there's no intentional changes to it that your body makes. So in general, all male descendants inherit the exact same Y-DNA with one exception. As it gets passed on, there's sometimes copy mistakes. And so those mistakes, which we call mutations, are the only reason that we get differences in Y-DNA from person to person. And they don't always happen. You know, you often will have Y-DNA passed on without any mistake. But then sometimes, every so often, you'll get a copy mistake in what gets passed on. But once those mistakes happen, those mistakes get, you know, passed on to the future generations. So descendants of the person that had that initial mistake all will inherit that same copy mistake. And so matches are basically determined on whether you have the same set of mutations in specific DNA locations that are known to have differences between all the men on the planet. So generally, Y-DNA, you know, you've got lots of similarity, and you're only checking a few specific locations to see if you have the mutations that are known to have happened in specific locations. Autosomal DNA, on the other hand, every person has 2 different versions of each chromosome. So you've got 1 from your father, 1 from your mother. And then with each child inherits a different patchwork of those 2 copies. And so it makes the, you know, brand new version for that child. And so the mistakes are actually only an insignificant aspect of autosomal DNA differences. The greater aspect has to do with who inherited what, and the mixing and matching of the 2 different versions that get passed on. So in autosomal DNA, it's not sufficient to just check a few isolated locations. Instead, you check, you know, continuous string of thousands of DNA locations and compare them. And if you've got a long enough continuous string of matching, then we assume that that's because you both inherited that segment from the same ancestor. So as an example, I've got, these are some names from my family tree. So these are my great-grandparents here, surnames, and then my grandparents, Clyde and Evelyn. So if we color the great-grandparents by different colors, and they each had a paternal and a maternal copy that got passed on, and in this case, I've got solid colors here, just I colored them based on what they inherited. So my grandfather had 2 copies, 1 from the Nisbet side, 1 from the House side. My grandmother had 1 copy from the Trout side, 1 copy from the Clevenger side, and then they had a couple of children. They each pass on a patchwork of their 2 sides, and so this is my I misspoke before. This is my grandfather actually, and my father is at the base of this tree, but. So Velton here has a different selection of the Nisbet and House copies than my Aunt E here, and a different selection of the yellow and green to her. So they both got a selection of those, but they each got a different selection. And then my dad would inherit a combination then of Velton would pass on a patchwork of his 2 copies, and then Joy, his mother, passes on a combination, and so his 2 copies then become those. And then the way matching works is if we were to compare, say, Keith with Aunt E here, the portions that if we compare all the colors there, the portions that they share here are, isolated there. That there's a little bit of green that both Keith and Aunt E have in one location. There's a little bit of red that both of them have. There's a little bit of orange that both of them have. A little bit of the yellow that both of them have. And so they've got these portions of shared DNA that they each have the same stretch of DNA. But the testing process, it turns out, doesn't distinguish between the two. You just basically get a sequence of what letters are present, or we call them letters. And so basically they can't distinguish what side is matching. It's just that there is matching in this location. So what the detected matching is going to basically be, in this case, I've got it black where there is matching and white where there is not matching. So you basically just get a sequence of, there's a stretch of matching here and then not matching there. The difficulty of autosomal DNA is then taking that matching that's detected and working out where it came from, because it's not directly labeled which one of the branches of the family it came from. It's just that you have some matching here. So what makes autosomal DNA challenging is that it's kind of a random jumble of bits and pieces of your ancestors all over your tree. It's not guaranteed to be from any specific branch of your tree. It can be from any branch of your tree. And so that means you have to actively investigate and identify and validate which branch each match comes from. They're not just automatic. So with Y-DNA, it comes from the direct paternal line. And so there's no question about where it came from. Whereas autosomal DNA, it's a puzzle that you have to find out. So many matches are going to come from unknown portions of your trees. Most of us don't know our trees fully out to 10 generations. And so there's significant chunks of our tree that we just don't know the ancestors that are in that portion of a tree, but yet you'll still get DNA matches that are from those portions of your tree, which makes it great for providing clues to genealogy, but it can also provide pitfalls in that you may have matches that come from portions of your tree that you don't know yet. It's also possible to have multiple, what we call most recent common ancestors with a match. So you may be related to some person via multiple different sets of ancestors. With Y-DNA, you've only got one direct paternal line, and so you're generally only going to have one connection with another match, whereas when you're dealing with all branches of your tree may intersect in multiple places with someone else's tree. So autosomal DNA in general requires very personal analysis. Every descendant gets a different selection of an ancestor's DNA, or you may not even get any at all if it's a distant enough ancestor. And so that makes it impossible to have a definitive, this is what, DNA from this line of Nisbets has, because every individual in the chain has a different collection of autosomal DNA. So generally autosomal DNA is associated with a specific person rather than from a whole line of people. So as an example, what we can do with autosomal DNA is basically create a DNA map. In this case, this is for my own DNA. So I've got 2 copies of every one of the chromosomes, and so my maternal and paternal. My paternal side is Nisbet and Blakeway surnames. My maternal side is the Gilchrist and Haining. And so I, have this initial map. And then if I were to, I've actually tested enough of my ancestors that I can fully map out to my, I guess this would be grandparents. So this is, I've now split in my paternal side into the red here is Nisbet and Trout, and then the blue is Blakeway and Huckel. And so you can kind of see that I've got different portions of the different chromosomes that are from different portions of my family. And so in my case, I've been able to identify which portions come from the Nisbet side versus the other portions of my family tree. But this is by testing a lot of different people and comparing where we match. But it gives you an example of kind of how you get this random patchwork of your different grandparents. And it works the same way if you continue going back further generations. So if I were to go another generation, I actually can map most of my great-grandparents on here. And so I added, it gets subdivided even further. And so you've got even more, just patchwork of various ancestors, and you can continue working your way back. I've got here just all the different generations, and the brighter colors are more distant generations, just because those are the ones that I find most interesting. And so you can basically create a map that then, if I have a unknown match that matches me in a specific chromosome location, I can go look up on my map and say, okay, this person is likely connected to me through this set of ancestors. And so that can be very useful if I know that they're connected to me through a specific line and they have that surname in their tree, then there's a pretty good chance that their person of that surname is connected to mine, and it can help focus our attention on trying to figure out how those are connected. So it can be very useful, but because it comes from all branches of your tree, it does take a lot of work to kind of map this out and leverage the usefulness of it. So how do we determine which branch our autosomal DNA came from? Each segment that you share with a match traces back to a most recent common ancestor shared by the two people. Basically, you each inherited that segment of DNA from some common ancestor. And so what you do is you take known matches and pay attention to which segments you share, because any matches that you share, match segments that you share with your known matches, those come from the known common ancestors of those of that person and you. So you search for your matches for people that you know or who have known relatives in their trees, or if you don't have any of those, you can also recruit your own known cousins to do DNA tests. And then you start building a map of those known segments. And so each segment where you know that, okay, with this person, our common ancestry traces through this ancestor, then you assign that segment to either the ancestral couple, if it's the normal, or I guess not normal, but the full relationship, or it may be a single person if it's a half cousin relationship. And then there are a couple of different third-party tools that you can use to help keep track of those segments. DNApanner.org is where the maps that I demonstrated come from. There's also a desktop software that you can use called genealogical DNA analysis software. And then you can, some people also will just use an Excel spreadsheet to keep notes on. So there's a lot of different ways that you can do this, but the main thing is if you're wanting to make use of this, you'll keep track of the segments of interest. The potential pitfalls of autosomal DNA is that you may have multiple intersections of your tree with the other person, and if only one of them is known, you may assume that that's where the match comes from, but you may have some other unknown common ancestor, possibly even more distant, that you weren't aware of, and so you didn't know to even be considering that could be from either source. So whenever we have a DNA match, we need to evaluate that the likelihood that we might have some unknown relative that is the source of the connection instead. If you have multiple DNA segments shared with a match, each one might be from a different ancestor. It's most of the time, all your segments will be from the same source, but that's not always the case. And so we just have to be wary of those assumption pitfalls. So to help with avoiding those, there's what's known as DNA triangulation. And that's based on the concept that everybody who shares the same DNA segment eventually converges on a single ancestor who's the source of that segment. If I only have me and one other person, there's still a reasonable probability that we might have a secondary source that we weren't aware of that we both share an ancestor from. If we get three people that all have different known descent paths from the same ancestor, then there's less chance that we all three also share a different common ancestor that we weren't aware of. So, and the more people involved in a triangulation, it's called a triangulation because we all three match each other. So I match, the person named John, and I match a person named Mary. Mary matches me and matches John, all on the same segment. So it kind of forms a triangulation of matching. But you can have more than three. So the more people involved, the better the confidence. If I can find, 15 people who all have different ancestral paths back to a single ancestor, then I have very high confidence because there's very little chance that you have 15 people that all share a second ancestor that they weren't aware of. So this is a process that you're basically just looking for as many matches as you can that all share the same DNA segment, and you can try to find the connection back to that same ancestor. So now we get into who should do autosomal DNA testing. Unlike Y DNA testing, it's equally valid for both male and female, so it doesn't matter whether you're male or female, it's equally valid. If you're interested in trying to look into a specific branch, like the Nisbet branch, then you're going to want to test the oldest generations possible along that branch. Every descending generation only inherits approximately half of what each of their parents had, and so you get less of that specific branch included in your DNA on average. So if you're targeting a specific branch, you want to find the oldest generations that are related to that branch in your family. Even if, so for example, I've tested some of my grandfather's siblings and cousins because they're still surviving, whereas my grandfather isn't. But they're the oldest generations on that branch of my family. Also, it's important to note that siblings each inherit a different selection of their parents' DNA. So if you can't go to an older generation, you can test multiple siblings to get a greater representation of what their parents had. With each additional sibling, you get an additional approximately 50% of what the other siblings didn't have. So it's a decreasing return with each additional sibling, but you're still in. So with one person, they represent approximately 50% of each parent. If you get two siblings, then between the two of them, they'll have around 75% of each parent. Three siblings, you're bumping up to 87.5%, and so on. So the more siblings you can test, the greater representation of what their parents would have had that you'll get, but it's decreasing returns. So once you have a good number of siblings, you're only going to get a little bit more with another sibling. And a similar concept with cousins. That just like each additional sibling represents more of their parents, each additional cousin will represent more of their grandparents. And so you can, if you don't have siblings, you could test, cousins to get additional representation that you don't. They'll have inherited some of your grandparents' DNA that you didn't have. If you have someone who already tested and you test one of their descendants, it doesn't add additional representation of that branch, but it may be of interest to for the other branches of that descendant. So if I test, one of my children, it wouldn't have any effect on, representation of my DNA, but it might provide a representation of my spouse's DNA, for example. If you're a participant in the Y DNA testing, then you probably already, have, deep DNA information on the Nisbet line. So doing an autosomal test probably isn't going to tell you anything more about the Nisbet line, but it might help others validate their connection to your Y line. So you'll have other people that may be descendants of a female Nisbet, so they don't have Y DNA, and they may not be certain whether they connect to your line or not. But if we can use autosomal DNA to show that they have a link via their Nesbit DNA to yours, then they would know that they connect to a specific Y DNA Nisbet line. So that even if you've already got Y DNA testing, it is helpful to others if you have autosomal DNA, because it can help them connect to a specific Y DNA line. As far as where to test, the Nesbitt-Nisbet project is only on family tree DNA, and that's because they're the only quality source of the in-depth Y DNA testing and matching, which, as we've seen earlier, is the one that's most associated with surnames. So it makes sense, if you're going to have a surname project, to have it at a place that has that in-depth Y DNA testing. So if you're wanting to do or contribute to the project with your autosomal DNA, you'll need to also have it on family tree DNA. They do accept uploads of raw data from other testing services, though, so you don't have to do your initial test with family tree DNA. We just need you to get your DNA onto family tree DNA. For autosomal DNA, the transfer results are perfectly acceptable for what we need to do. So wherever you choose to test initially, as long as they offer a download of the raw data that you can then upload to Family Tree DNA, that will be perfectly fine. It's free to transfer and see the match list, but it costs $19 on Family Tree DNA to unlock the chromosome browser. And that's what we use to see the specific segments that match. And that's, as we've seen before, that is very important for validating that a specific match is from a specific branch of your family. So if you're trying to be useful to the Nesbitt-Nisbet project and you're transferring from elsewhere, you would need to go ahead and pay that $19 to unlock the chromosome browser, because otherwise it's not as useful to us. So you can either choose to test directly with Family Tree DNA or test from an alternate site and transfer your results to Family Tree DNA. The other major sources that are known to be reputable DNA testing sites are 23andMe, Ancestry DNA, and MyHeritage. There are a few others that are smaller services that would also work. But if you're just wanting to get into DNA testing, these are the three big ones that I would say are the ones you're choosing from if you're not wanting to do Family Tree DNA as your initial testing. Just kind of an overview of the comparison between them. Family Tree DNA, as I mentioned, is the only one that has in-depth Y DNA matching. And so that's why the project is with them. They actually have a relatively small autosomal user database. So that may be one reason why you might want to test elsewhere first and then transfer in to contribute to the project. But they do have autosomal DNA testing. They've got a set of all the basic autosomal analysis tools. They don't have a lot of automated hints that some of the other sites have to help you try to figure out what your common ancestry is. They basically assume that if you're doing this, you're into DNA aspects, and so you're going to communicate with your matches to try to figure things out. So they don't try to do any of your work for you. They're also only focused on DNA. They don't have non-DNA genealogy tools on the site. Everything that they have on the site is solely about DNA. MyHeritage is a relatively newer company, so it still has a relatively small user database. They're working on trying to grow it, but at this point it is one of the smaller user database. And the key thing with the size of the user database is if you've got a small user database, you're going to have a smaller number of matches, which means less opportunities to try to find clues and make use of the matching segments to map out your DNA. The MyHeritage does have a good selection of automated tools, and they also have a subscription service to general genealogy tools apart from DNA, and they will also accept uploads from other services. So a good all-around selection of tools available from them. The downside of them at this point is just that they're newer, so they've got a smaller user database, and some of their tools, they're still working on trying to refine them. But long-term, I anticipate they'll be a pretty solid company. Ancestry DNA is one of the older ones around, so they have by far the largest user database of autosomal users. They don't have the Y DNA. They're only autosomal DNA, but if you're looking at autosomal DNA, you're likely to get the most matches from Ancestry DNA. They're also well known for their just general genealogy tools that they have subscription services. So if you're just into genealogy in general, they're a major player in that field. They also have a good selection of automated tools for helping you, figure out what your shared ancestors with your matches are. So that can kind of help ease a little bit of your workload. But they also don't have the chromosome browser to check the specific location of your match segments. They also do not allow uploads from other services. So if you're going to get into their database, you have to test with them. So if you're into genealogy in general, they're a big one that a lot of people will start with and then transfer to Family Tree DNA and or MyHeritage. And the main reason they don't do the chromosome browser is that they feel that it could lead to privacy issues as far as if someone they share a match with you on a specific chromosome location that's associated with some health risk, then they might know something about you that you don't want them to know. For me, that isn't an issue that I'm concerned with, but it's just something to be aware of. That's the reason that they don't provide the chromosome browser. But it does make it harder to validate that a match comes from a specific branch of your family. They tend to get around that by having such a large user database that just by sheer numbers, you can often assume where a match comes from because you've got so many matches from that branch of your family instead. 23andMe is the final option here. They tend to focus more on health aspects than any of these other sites. So they've got a lot of tools that will kind of screen your DNA for whether you might have health risks on specific things. So if you're interested in that, that can be a site that people choose to start with. They are also focused on DNA. They don't have just general genealogy tools. All the tools that they have are DNA focused. They also do not allow uploads from other services. So if you want to be in their database and use their services, you have to test with them. So that's kind of an overview of the different major players. Which one you choose to start with, it kind of just depends on what interests you, whether you're more interested in the genealogy side of things or the health side of things, or you just want the simplicity of directly testing with family tree DNA to contribute to the project, and you're not interested in pursuing any of the other things on your own. So it's kind of up to you. The main thing that we need for our project is eventually upload to Family Tree DNA and join the project. Then as far as how to make your test most useful, attach a tree to your Family Tree DNA account. As we've mentioned, the autosomal DNA can come from any portion of your tree, and so it's important to be able to check what your tree is against a matches tree in order to check what the likelihood there is that there is a secondary shared ancestor that we aren't aware of, because it's very easy to make assumptions that a match is from some known common ancestor, but completely miss that there's a second or third possibility. So having the tree available to us very significantly will help. Also, you want to ensure that your kit settings give the Nesbitt-Nisbet project administrators very minimum the limited access level. Advanced is what gives us the most access, so it's preferred from our end. But if you're concerned about privacy, or if you think that the Nesbitt administrators are going to try to take too much control, I think for the Y-DNA, they generally always recommend advanced level of access. For what I need for the autosomal, limited is sufficient for what I need to do. If you're wanting specific help with it, the advanced gives me a little bit more access. But the minimum option on that blocks access to your tree and matches, which are very important to doing autosomal DNA. So if it's going to be useful to us, we need to have at least one of these other two settings. Also, when granting admin access, it's often useful to select the future administrator to make sure that if the administration of the project gets moved over to a new administrator, that they still have access to make use of your DNA. Also, it's recommended to set a kit beneficiary that beyond your lifetime, it'd be useful to still have your DNA results contributing to the project. So having a kit beneficiary ensures that that doesn't get blocked from us being able to use it. And then apart from Family Tree DNA, it's also useful to make sure that you and your ancestors are added to the wikitree.com tree. That's basically a worldwide tree where everyone is working on the exact same tree. So it's a group effort. And our coordinator, Gene Starr, has been leading an effort to try to make Wikitree a public location where we keep the most recent Nesbitt-Nisbet genealogy knowledge and make that centralized and available to everyone. And the advantages of doing it this way is that if we find new information that, some theorized link between two people is disproven by DNA, we can get that on the tree and show the proof of it and then it gets out everywhere, rather than having everybody have their own tree, and misinformation that gets spread out there is a lot harder to correct. So having a centralized tree where we can have the most up-to-date information is, I think, very beneficial. So making sure that you and your tree are part of that are great. You can also then add your DNA kit info, and what that does is allows anybody else researching related branches to know that your test exists, and maybe your test could help them prove whether or not they're connected to your branch, or if there's maybe somewhere in there where the biological relationships don't match the documented relationships, then the DNA test may be able to help validate or disprove a connection. So knowing which tests are out there and exist are useful to researchers. So that's just something generally that's useful to go ahead and add yourself to the wikitree.com tree, and then add your DNA kit info so that people know that it exists. And then importantly, also recruit your Nisbet relatives to test and join the project. As we've seen, autosomal DNA is most useful if you have a lot of known relatives that can help you identify and map out which portions of your DNA is from the relevant branches that we're trying to pursue. So the more we get involved, the more we can make use of it. And that's something that actually the autosomal side of our Nesbitt-Nisbet project, at this point, we're pretty much purely in just recruitment phase. It's likely to be a while before we get enough participation to really be able to make use of it. It's somewhat of an experimental project at this point. I'm here to answer questions and help people understand their autosomal DNA, but we're hoping that eventually we'll get enough participation that we can start using it to link people with their Y-DNA lines that maybe they weren't aware of the connection to. So we're hoping to be able to make use of it in the future. But autosomal DNA, because it comes from all sorts of branches and it takes so much intermatching to be able to identify which branches it comes from, it's very important that we have a sufficient participation base in order to be able to make use of it. So it's probably going to be a little while before we are able to up the participation in the autosomal side of things to the point where it can actually be used. This is just kind of on the WikiTree quick image here to show where you can find on the DNA test information. You can add new tests, and then you can add tests from a lot of different places. So depending on where you were initially tested, you can add it. And then it basically, so for example, on Ancestry, you indicate your username, and you can also provide any additional information. And then that just lets people know where to go looking. If they want to be able to match you and test to see if there is a matching connection, which site they would need to test with. So if you have added your DNA to multiple websites, then it's useful to go ahead and add all the ones that you've added it to, so they know where to find it. And with that, that kind of wraps up what I have to say today. If you have any further questions, I've got contact info here for both Gene Scar, who's the Society DNA coordinator and the specialist in the Y-DNA side of things. I am the specialist on the autosomal DNA side of things. So if you have questions specifically about autosomal DNA, you can direct them to me. And with that, I will close the presentation. Thank you.